Thank you, Yang. Next, I would like to continue the second part of this presentation. I will introduce how the PBPK modeling can serve as a part of the totality of the evidence to support approval of the mesalamine DR tablets. Here is my disclaimer. And learning objective, for my part, I will mainly explain how to develop PBPK model to predict a local drug amount by incorporating dissolution data at different pH. First, we want also to identify which dissolution is the biopredictive. A little bit of background on the GI locally acting products. As we know, assessing the BE for GI locally acting products is challenging because the systemic exposure may not reflect drug concentration at the site of action. So the BE recommendations of oral locally acting drug products are based on the drug product properties and mechanism of action. PBPK models may be used to explore the correlation between dissolution in GI tract and the plasma concentration profiles and predict the local drug amount and support BE evaluation of such products. As Yang mentioned about these background of the mesalamine DR tablets, it is indicated for colitis. And the product-specific guidance for this product recommends fasting and the fat PKBE study, and also the comparative dissolution studies at different pH, of course, include the pretreatment stage 1 at pH 1.2 and pretreatment stage 2 at pH 6.4, and then evaluate the comparability at each different pH, 6.5, 6.8, 7.2, and 7.5. However, for this generic drug product, we found the F2 values less than 50 when comparing the dissolution between test and reference at pH 6.8 and 6.9. However, it's more than 50. It's comparable when the dissolution is over pH 7. But the test product and ROD were found to be bioequivalent for systemic PK under both fasting and fat conditions. Here comes the question, whether the slower release of test product at a certain pH would significantly impact the local drug exposure compared to ROD. And you have seen these PSG recommendation and know the conditions we recommended for conducting the in vitro comparative dissolution study. And in this, for this generic drug product, we found that at pH 6.5, 6.8, 6.9, the dissolution are not similar between test and reference, and the F2 is less than 50. You know that we use these in vitro dissolution testing over a range of pH to serve as a surrogate of in vivo drug release in the GI tract. And we see actually these test product shows decreased release compared to ROD at pH less than 7, but it's comparable at pH over 7. Whether these will cause any significant impact on the BE for this local drug, GI locally acting products, we will use PBPK modeling to support providing some evidence. And we use the PBPK modeling to try to identify which dissolution condition is biopredictive. And we will also use PBPK modeling to predict the local drug amount in the colon and use population simulation to predict the percentage of drug absorbed in the colon. So the so here I would like to use a little bit of this flow chart to go over the how we develop, validate, and apply this PBPK model. We first use the intravenous PK data obtained from the literature to estimate disposition parameters. Then we develop the PBPK model for mesalamine DR tablet. The intestine first pass effect was optimized. And then we also incorporate 3-stage dissolution data with pH 6.8, 6.97, and 7.2 as stage 3. Incorporate them into PBPK model and predict the PK, systemic PK. And then the PBPK model was validated using dissolution and the PK data obtained from literature or other ANDAs, and to validate this model. And then after validation, we use this PBPK model to see which which dissolution is biopredictive and predict the drug amount in the colon. These two figures shows that PBPK model can capture the PK well after IV bolus administration of 250 mg mesalamine. And it can capture the PK after oral administration of the delayed release tablet. And we use other data from literature, and we can also see the modeling predict well of the PK. And we, as shown on the right table, we use the dissolution data and the PK data obtained from the other ANDAs, and we show that the PK prediction actually is pretty well, and with the prediction error less than 20%. This means that the model is validated using these additional data. Then we use the validated data to evaluate whether the dissolution data at a certain pH is biopredictive. As shown in these tables, we can see if we use the dissolution at pH 7.2 and 7.0 as stage 3 incorporated into the PBPK model, then the prediction error is less than 22%, which means these conditions are biopredictive. But if we use the dissolution at pH 6.8 as stage 3 into the model, then we cannot predict PK well with the prediction error more than 30%. So we know that the dissolution at pH 7 and 7.2 at stage 3 are biopredictive. So then we incorporate this biopredictive dissolution into the model and predict the local drug amount in the colon and also the percentage drug absorbed in the colon. As you can see from the right top table, and the simulation suggests that the local amount in colon is similar between R and the test product, ROD and the test product. And the population simulations showed that percentage of drug absorbed in the colon is similar between the ROD and the test product, with 90% confidence interval of this TR ratio for the drug absorbed in the percentage of drug absorbed in the colon can fall within 80 to 125. This should demonstrate that there is a low risk of non-BE at this site of action for the generic drug product. These two figures you have seen previously with Dr. Yang Lu's presentation, and based on the formulation design, release mechanism, and GI physiology, shown here, we know actually the drug, the generic, both RD and the generic drug, they have the enteric coating. The coating will be break down after it enters the in the small intestine, especially the terminal ileum, where the pH is 7. Then the core continues to pass into the GI tract and go to the colon and continues to release the drug. The colon, you know that the pH is around 6 to 6.7. And we can also see the drug, actually, in the scintigraphic figure, the drug release in the ascending column, it's at the ileum, terminal ileum. It's kind of consistent with what we are thinking about the release mechanism. These are also consistent with our PBPK modeling. And the PBPK modeling shows that the dissolution at pH over 7 is biopredictive, but not for the dissolution at pH 6.8, because the drug is already break down before it enters into the colon. So the pH 7 is more biopredictive, as demonstrated by PBPK modeling and also the release mechanism. So in summary, the PBPK modeling simulations supported that the 3-stage dissolution profiles at both pH 7, 7.2 at stage 3 may be biopredictive to the local and systemic exposure. The predicted amount of mechanism in colon and the percentage of drug absorbed in the colon, they are similar between ROD and the test product. So the totality of evidence based on the dissolution data, formulation differences, gut physiology considerations, along with the PBPK modeling results were used to conclude that this generic drug product has low risk of bioinequivalence at site of action compared to the ROD. So this work, as mentioned by Dr. Yang Lu, supports the approval of this mesalamine generic drug product. So here comes a challenge question. To evaluate the BE for locally acting products, the evidence could include the following. A, formulation and the dissolution comparison. B, gut physiology considerations and release mechanism. C, PBPK modeling and simulations. D, all of the above. I will let you think about 1 minute. Yeah, 1 second. Then the answer is all of that, considering the time limitation. With that, I would like to acknowledge our colleagues in DQMM and also the reviewers from Office of Bioequivalence. Especially Dr. Sharon Thomas, who did most of the modeling work, and the reviewers from OB, Dr. Yang Lu, Dr. Teresa Lee, and Gong, and they are valuable inputs and collaboration during the review of this generic drug product. Thank you. Then I will turn over to Dr. Li Kan Liang. Thank you so much.